Every second, the male body produces about 1,500 sperm. That's over 100 million a day. But the quality of those sperm can mean the difference between fatherhood and infertility. And it's influenced by your brain, hormones, lifestyle, and yes, your sexual habits. In fact, male fertility doesn't just determine the chances of conception. It can influence the genetic health, resilience, and long-term well-being of your future children. In this video, we'll explore how sperm is created, the fascinating brain-body connection behind it, the hormones that control the process, and how lifestyle factors can either strengthen or weaken fertility. We'll also examine the science of masturbation versus real intercourse and their impacts on sperm quality. And finally, we'll separate the myths from the facts, giving you practical tips to improve male reproductive health. The male reproductive system is a highly coordinated network of organs and hormones working together to produce, mature, and deliver sperm. The testicles, located in the scrotum, are the primary production site. Inside them, millions of tiny tubules are dedicated to making sperm cells. Once produced, sperm travel to the epididymis, a long, coiled tube where they mature and gain the ability to swim. From there, they move through the vas deferens, the transport highway leading toward the urethra. Along the way, fluids from the seminal vesicles and the prostate gland mix with the sperm to form semen, providing nourishment and protection for the journey ahead. This entire process is guided by a hormonal control system starting in the brain. The hypothalamus releases gonadotropin-releasing hormone, or GnRH, which signals the pituitary gland to release luteinizing hormone, LH, and follicle-stimulating hormone, FSH. LH stimulates the Leydig cells in the testicles to produce testosterone, the primary male sex hormone, while FSH works with testosterone to drive sperm production. Another hormone, inhibin, acts as a feedback signal to the brain to regulate production and maintain balance. Sperm production, known as spermatogenesis, is not instantaneous. It takes about 64 to 72 days for a sperm cell to fully develop from its earliest stage to a mature, motile form. This means that the quality of your sperm today is a reflection of your health, nutrition, and lifestyle choices from the past 2 to 3 months. Every decision you make during that period, from sleep to stress management to diet, leaves its imprint on your reproductive blueprint. Sperm creation begins deep inside the testicles in structures called seminiferous tubules. Within these microscopic tubes, the process starts with germ cells. These are the body's raw reproductive stem cells, and under the right hormonal signals, they develop into spermatogonia, the first identifiable stage of a sperm cell's life. Once spermatogonia are formed, they begin a series of divisions and transformations. One of the most critical steps is meiosis. Meiosis is a special kind of cell division in which the genetic material is reduced by half. This means each sperm will contain only 23 chromosomes. The reason is simple but vital. When a sperm and an egg combine, their chromosomes add up to the complete set of 46 needed for a healthy human being. Without meiosis, reproduction would not work as intended. After meiosis, the developing sperm cells enter their maturation phase. At this point, they start transforming into spermatozoa, fully structured sperm cells with a head, midpiece, and tail. But they are not yet ready to function. During this stage, they are supported by Sertoli cells, which act like caregivers and protectors. Sertoli cells supply nutrients, manage waste removal, and form the blood-testis barrier. This barrier is crucial because it shields developing sperm from harmful substances and immune system attacks that could damage them before they are complete. When the spermatozoa are fully formed, they are still immature in terms of movement. They travel to the epididymis, a long, coiled tube attached to the back of each testicle. The epididymis is like a training camp where sperm spend several days learning how to swim efficiently and gain the stamina they will need for the journey toward the egg. This is also where sperm undergo final quality checks, with defective or poorly shaped sperm removed from the lineup. 3 qualities largely determine a man's fertility potential. The first is sperm morphology, the shape and structure of the sperm. The second is motility, the ability to move strongly and in the right direction. The third is count, the total number of sperm produced. A high count of well-shaped, highly motile sperm gives the best chance for successful fertilization. Any weakness in these 3 areas can significantly reduce the likelihood of conception. One fascinating detail lies in the sperm's tail, called the flagellum. At the base of the tail is the midpiece, which is loaded with mitochondria. These mitochondria are the power plants of the sperm, producing the energy needed for the tail to whip back and forth. This constant motion propels the sperm forward, sometimes across enormous distances in microscopic terms, to reach the egg. From a single germ cell to a highly specialized swimmer, sperm production is a process that takes roughly 70 days. It is a precise, coordinated effort between cells, hormones, and structures, and every step plays a critical role in determining male fertility. At the center of male fertility lies a communication network between the brain and the reproductive organs, called the hypothalamic pituitary gonadal axis, or HPG axis. It works like a chain of command. The hypothalamus, a small region deep in the brain, sends out signals in the form of a hormone called gonadotropin-releasing hormone, or GnRH. This hormone stimulates the pituitary gland, located just below the brain, to release two more hormones, luteinizing hormone and follicle-stimulating hormone. Luteinizing hormone travels to the testicles and tells the Leydig cells to make testosterone, while follicle-stimulating hormone works together with testosterone to drive sperm production inside the seminiferous tubules. This loop is carefully balanced. Too little signal and fertility drops. Too much and the system shuts down through feedback. Testosterone has a dual role. On one side, it is essential for spermatogenesis, making it the key hormone of male fertility. On the other side, it shapes the physical and behavioral traits we associate with masculinity: muscle growth, deeper voice, facial hair, and sex drive. Without enough testosterone, both fertility and those traits begin to weaken. But this delicate axis can be disrupted. Stress, depression, and lack of sleep are among the strongest negative influences. When the brain perceives stress, it increases cortisol, the stress hormone. High cortisol interferes with the hypothalamus and pituitary, reducing the release of GnRH, LH, and FSH. As a result, testosterone levels fall and sperm production slows down. Poor sleep has a similar effect because testosterone is produced in pulses during deep sleep. Chronic sleep loss, ongoing stress, and mental health struggles can all combine to reduce sperm count and lower fertility over time. The brain's influence does not stop at hormones. Neurotransmitters released during sexual activity also play a role. Dopamine, the reward chemical, rises with sexual arousal and orgasm, reinforcing the desire to engage in sex again. Oxytocin, often called the bonding hormone, is released in higher amounts during partnered sex compared to solo activity. This difference may have evolutionary roots, strengthening pair bonding and cooperative parenting. In solo sexual activity, dopamine release still occurs, but oxytocin release is significantly lower, which means the brain and body experience the event differently. These subtle changes in brain chemistry may not directly harm fertility, but they shape the overall connection between sexual behavior, hormones, and reproductive health. The HPG axis is therefore not just a chain of glands and hormones. It is a reflection of the brain's state, mental health, and social behavior. When the brain is balanced, rested, and supported, fertility thrives. When it is stressed, deprived, or isolated, the signals falter and reproductive health begins to decline. When it comes to male fertility, many people wonder if there's a real difference between solo sexual activity and partnered sex. Both end in the same basic outcome, ejaculation, and both trigger similar short-term chemical changes in the brain and body. During orgasm, whether through masturbation or intercourse, dopamine surges to create a sense of reward, prolactin rises to trigger the refractory period, and the nervous system shifts from high arousal back into a recovery state. In this way, the mechanics of climax look very similar. But science also shows clear differences. Partnered sex consistently leads to a stronger release of oxytocin, the hormone linked to bonding, trust, and pair connection. Masturbation produces only a small rise in oxytocin, while intercourse causes a much more pronounced release, reinforcing human bonding and long-term social connection. Another difference comes from testosterone. Research suggests that sexual arousal with a partner can cause a temporary rise in testosterone levels, higher than what is usually seen during solo stimulation. This may provide a short-term boost to sexual drive, energy, and even sperm function. There is also a physical difference in how the reproductive tract responds. During intercourse, the orgasm often involves stronger muscular contractions, especially in the pelvic floor and reproductive ducts. These deeper contractions may help sperm move more efficiently through the reproductive tract, improving the chances of sperm transport and delivery. Solo activity also produces contractions, but usually not with the same intensity. Now let's address sperm health and ejaculation frequency. Ejaculation is the body's natural way of renewing sperm, and frequency has different short-term and long-term effects. If a man ejaculates daily, the sperm count per sample may temporarily decrease because the body has less time to build up large numbers between releases. However, this does not damage fertility in the long run because production continues at a steady rate. On the other hand, long periods of abstinence beyond about 7 days tend to increase sperm count per sample, but those sperm may lose motility, which is their ability to swim effectively. In fertility terms, a balance is usually best, not too frequent to reduce concentration and not too infrequent to reduce quality. This brings us to one of the biggest myths, the idea that masturbation causes infertility. The reality is that it does not. Normal levels of masturbation have no negative effect on long-term sperm production or overall fertility. What matters far more are lifestyle factors such as stress, sleep, nutrition, alcohol, smoking, and environmental toxins. Those elements can impair sperm count, motility, and morphology, but masturbation itself is not harmful. In fact, regular ejaculation can support sperm health by clearing out older, less functional sperm and making space for newer, healthier ones to take their place. So, while both masturbation and intercourse share common ground, intercourse provides unique hormonal and physical benefits, especially through oxytocin bonding, temporary testosterone boosts, and stronger contractions that aid sperm movement. Masturbation is not harmful, but it is not a substitute for the complex brain-body social feedback that partnered sex provides. The takeaway is simple. Male fertility is not destroyed by masturbation, and it can even be supported by regular ejaculation. But the hormonal and emotional dimensions of intercourse add an extra layer of benefit that solo activity does not fully replicate. Fertility, in the end, is shaped by the combination of biology, brain chemistry, and lifestyle, not by myths. Male fertility is not just about anatomy and hormones. It is powerfully shaped by the choices you make every single day, and science shows that nutrition, exercise, body composition, heat exposure, substance use, and sleep can all determine how well sperm are produced and how healthy they are. Let's start with nutrition. Certain micronutrients are absolutely essential for sperm production and function. Zinc is required for the enzymes that allow sperm to mature and fertilize an egg. Selenium helps protect sperm from oxidative damage, which is critical because sperm membranes are very delicate. Vitamin D is linked to testosterone production and improved sperm motility. Antioxidants such as vitamin C and vitamin E protect sperm DNA from free radical damage. Diets rich in whole foods, fruits, vegetables, lean proteins, and healthy fats are consistently associated with higher sperm counts and better morphology. Now, exercise. Moderate resistance training has a clear benefit. It stimulates testosterone production, improves circulation, and supports overall metabolic health. But overtraining tells a different story. Excessive exercise combined with inadequate recovery elevates stress hormones such as cortisol, which can suppress the hypothalamic pituitary gonadal axis and lower testosterone. The balance between activity and recovery is the key. Body fat is another important factor. Both extremes, too high or too low, impair fertility. High body fat is linked to reduced testosterone, higher estrogen, and lower sperm count. On the other hand, very low body fat, often seen in extreme athletes, can also suppress hormone production and interfere with normal spermatogenesis. The healthiest range is a moderate body fat level where energy balance, hormones, and nutrient availability all align. Heat is often underestimated but plays a real role. Sperm development requires a slightly cooler temperature than the core body. Anything that increases testicular heat for long periods can damage sperm. Tight underwear, frequent hot baths, saunas, or placing laptops directly on the lap can raise scrotal temperature and reduce both sperm count and motility. Even small increases in temperature, sustained over time, can have measurable effects. Substance use also makes a strong impact. Alcohol in excess disrupts testosterone production and damages sperm DNA. Smoking introduces toxins that increase oxidative stress, lowering motility and morphology. Anabolic steroids may cause a short-term boost in muscle mass, but they almost always suppress natural testosterone and can completely shut down sperm production. Cannabis and certain medications also interfere with the hormonal balance required for fertility. Every one of these substances can leave a measurable fingerprint on male reproductive health. Finally, sleep. Sleep is not optional for fertility. Even a single week of restricted sleep can cause testosterone levels to fall significantly, impairing sperm production. Deep, consistent sleep supports the hypothalamic pituitary gonadal axis, stabilizes hormone release, and gives the testes the recovery window they need to sustain sperm creation. Poor sleep is one of the fastest ways to reduce fertility, and good sleep is one of the simplest ways to restore it. In short, lifestyle is not just background noise. It is a central player in male reproductive health. Nutrition fuels the process. Exercise strengthens it. Body composition provides the foundation. Heat and toxins disrupt it, and sleep protects it. Every choice you make leaves an imprint on your fertility and ultimately on the genetic health you pass on to your children. When things go wrong. Even with a healthy lifestyle and normal anatomy, fertility challenges can still appear, and it is important to understand the common causes of male infertility. One of the most frequent issues is varicocele. This is an enlargement of the veins within the scrotum. It can raise testicular temperature, reduce oxygen supply, and impair sperm production and motility. Varicoceles are treatable, but if left unchecked, they can make conception more difficult. Low testosterone, also called hypogonadism, is another cause. Without enough testosterone, the process of spermatogenesis slows down. Men may also experience low energy, reduced muscle mass, and low libido. Treating the root cause of low testosterone can restore both hormones and fertility. Genetic factors also play a role. Conditions such as Klinefelter syndrome, where an extra X chromosome is present, often reduce or completely block sperm production. Y chromosome microdeletions are another hidden cause where key genetic regions required for spermatogenesis are missing. These cases often require medical intervention or assisted reproductive techniques. Infections and blockages are also important contributors. Past infections in the reproductive tract, scar tissue, or congenital blockages can prevent sperm from traveling through the vas deferens or urethra, even if sperm are being produced normally in the testes. So how do you know when to seek help? A semen analysis is the first step. This simple test measures sperm count, motility, and morphology. If a couple has been trying to conceive for a year without success, or 6 months if the female partner is over 35, it is time to get checked. For men, a semen analysis can quickly reveal whether sperm health is within normal ranges or if further testing is required. Understanding these causes is not about fear, but about clarity. Many cases of male infertility are diagnosable and treatable, and early testing often makes the difference between years of uncertainty and a clear path forward. Improving fertility is not only about medical treatment. Everyday lifestyle choices make a real difference. First, focus on a balanced diet rich in zinc, selenium, vitamin D, and antioxidants like vitamin C and E. These nutrients support sperm quality, and supplements can be added if the diet is lacking. Keep the testicles cool. Sperm production is very sensitive to heat, so avoid tight underwear, prolonged hot baths, or keeping a laptop directly on your lap. Cutting down on alcohol and smoking is critical. Both directly damage sperm DNA and reduce testosterone levels. The same applies to recreational drugs and anabolic steroids. Managing stress is equally important. Chronic stress disrupts the brain's hormonal control of fertility and lowers testosterone. Relaxation techniques, mindfulness, and quality downtime can restore balance. Maintaining a healthy weight and regular exercise routine is another key factor. Moderate resistance training boosts testosterone and sperm health, but overtraining can have the opposite effect. And finally, regular ejaculation can help reduce DNA damage in sperm by clearing out older cells and encouraging fresher production. This does not harm long-term fertility and may actually improve overall sperm quality. Simple, consistent habits like these create the strongest foundation for male reproductive health and future fertility. In conclusion, remember this simple truth. Sperm health is male health. The quality of your sperm reflects the state of your hormones, your metabolism, your stress levels, and even your daily habits. By taking care of your body through nutrition, exercise, sleep, and balanced lifestyle choices, you are not only protecting your fertility, but also strengthening your overall health. Male fertility is not just about reproduction. It is also a marker of long-term vitality and even the future health potential of your children. And this is just one side of the story. In an upcoming video, we will explore how female fertility works and how both male and female factors come together in reproduction. If you found this breakdown useful, make sure you subscribe to the channel so you don't miss that episode and many more science-based deep dives into health and human biology.